Good afternoon. Welcome to the webinar, Good Bugs, Bad Bugs. Thank you for joining us. Our speaker is Dr. John Skinner, Professor, Extension Apiculturist, and Coordinator at the University of Tennessee. Dr. Skinner's bee-related research includes IPM of honeybee pests, improving habitat for pollinators, cooperating with a bee-informed partnership, and I haven't read how many hives are included in his research, but I'm betting there are several. After a couple of introductory slides, Dr. Skinner will present his program. At the end of his program, and a couple of closing slides, and a time for questions and answers, you'll be asked to participate in a very short poll. For those with 5 extra minutes, there is an additional brief survey. The honeybees would greatly appreciate your response to it as well. My name is Sally Lee. I'm in Birmingham, Alabama. Welcome to you. And let me give you one more slide. Where have all the honeybees gone? Hope for the future. Dr. Skinner.
Thank you very much, Sally, and welcome to the webinar today. The topic I'm dealing with is actually a very serious one. Since everybody has heard lately in the news the challenges we're having with honeybees, where have they all gone? So today I'd like to look at this issue with you, explain some of these challenges, give you a better understanding of what is actually happening. And then I will also lead this to what we're doing about this. How can we prevent this loss of bee pollinators that are so important to our agriculture? Why do we need bees? Well, it's pretty obvious to most people. A very simple way to say it is if you like to eat, you need bees. About every third bite you put in your mouth is a direct result of bee-pollinated crops. Either they must be pollinated to set a fruit, or it greatly increases their value of the entire crop. This is huge throughout the world, throughout the United States, and probably in your backyard and your home gardens as well. All those things in the baskets require pollination, either to make the fruit or produce a seed that is itself a seed crop, so those packets of seeds can go out to gardeners and they can produce their own in their own gardens. I bet most of you have gardens, and that's maybe why you're tuned in here. Thanks for being here. What about the economics? Economics always say a lot. In the United States, we've got over $25 billion crop value annually for crops that benefit from bee pollination substantially. In Tennessee, we're probably in excess of $500 million in our crops. In your own state, you probably have similar things. The picture shows you colonies of honeybees that are in almonds in California. This particular crop requires well over 1.25 million colonies of bees to set that almond crop. So think about that the next time you munch on one of those crunchy nuts. They're also good for you too. A while back, and I want to say 2006, 2007, there were severe challenges going on, and it was labeled at that time colony collapse disorder. Well, what is this? What is this situation? You notice it's a disorder. It's not a particular causative agent. It's not called a disease. Well, what could be some of the causes? We know that mites are involved. These are parasitic mites. They're serious pests of honeybees. We know that there are some diseases in there that cause all kinds of symptoms in a hive that are a red flag to that beekeeper as they're opening their hive and they know that something is going on. Could this be due to pesticide contamination? Both due to chemicals that we beekeepers put in our colonies to combat these mites, as well as other things that are outside in the environment that we're spraying on in our gardens and treating our seed with. Could be beetles, could be, what about nutrition? What about this as a factor? Beetles, probably not so much. They may be a local problem. Nutrition, yes. It's now emerging as a major factor, okay? But this whole thing led to some serious work. And also reporting initially, you can see the areas on your screen that are colored yellow are areas that were confirmed that had CCD. However, some of this may be a little bit exaggerated because only one beekeeper from an individual state had to call up to get their state colored in. And that beekeeper may or may not have understood exactly what CCD was. I'm cautious when I talk to beekeepers that they don't use CCD as a scapegoat for not doing correct kind of beekeeping. But certainly they wouldn't do that, would they? Okay, we have had declines in number of beehives through the years. And we certainly are aware of this situation, and a lot of it had to do with parasitic mites, including the tracheal mite in the late 80s, early 90s, and varroa mites. But let's get back to this colony collapse disorder. Why was it called anything at all? Well, it's based on symptoms that are seen inside the beehive. This is the home of the bees, where they live, where they raise more bees. They go through this whole brood cycle. That's just a term for the collective development of bees from egg to larva to pupa to adult. We found that bees would be failing to return to the hive. What did this result in? Well, you really didn't see dead bees lying all over like you might expect for a pesticide incident or something like that. There were small clusters of young adults and the queen, and the colony losses were really rapid. And if you look at the frames below held by the beekeeper, the one on your immediate left is pretty well covered. That brood is covered over and incubated at 95 degrees plus or minus 5 to have healthy brood development and those pupa underneath those brown cells to mature and develop correctly. The one in the middle, though, is not covered all the way. These are symptoms we'd see. We'd see, where are the adult bees? They're not covering the brood frames. The ratio of the amount of coverage of bees and brood flip-flopped. It went from a normal pattern on the left of 2 times as many surface area of bees to 1 surface area of brood to the reverse. When you looked in there, the brood was exposed, and therefore that brood was dying because they weren't incubating it at the correct temperature. There were others of us that as scientists, we have to be inquisitive. We have to decide, are these colony losses similar to a period before CCD? We've had this decline due to a number of factors. Is this an exaggerated problem? Maybe. Are there other episodes in past history where we've had diseases that went through and we didn't know what it was? Well, certainly we had what was called disappearing disease that later someone discovered it was actually probably due to tracheal mites. Who would have thought to look in the tracheal tubes? Beekeepers just saw colonies dying. They seemed to get better on their own, and then another 10 years later, we saw it again. So what is this? Is it something brand new? Are these new symptoms, a new pest, new pathogen? Well, we really didn't know, but we knew we needed to do something about it. And one benefit that it has had, it did stimulate new studies. And we determined after a lot of work that CCD was just one aspect of a much bigger picture that we're calling honeybee decline. So what's being done about CCD, and what did we do as a group of people? Well, we established a lot of working groups pretty fast, put our heads together to try to come up with solutions to these problems. What was a regional group? Basically NC 217. This led to a more formal group and included 35 participants, 23 different states, USDA, state universities, agencies. We put our heads together, and this was important. It resulted in a coordinated agricultural project, a CAP, protection of managed bee colonies. And we received $4.1 million in grant to try to figure out some of this problem. We set these objectives. It's a lot of words on a slide, so bear with me. To determine the cause of it. It's complicated. We knew that. It wasn't something that just we could take a quick look at and say, oop, that's it. Could it involve breeding? We know that genetics underlie all kinds of activities and the health of the bees. And bee genetics are complicated because they multiple mate. And that's part of a challenge. Are our genetic stocks becoming too inbred? We were concerned about that. We knew we needed to conserve non-apis, non-honeybee pollinators, because if we lost the bees, what would pollinate some of our crops? So we felt that was important. And we knew that whatever we discover in these things needs to be translated in a language that beekeepers and growers can understand as well as scientists. So the extension component was very important here. And we were very involved in that aspect because they put me in charge of part of that extension component. What were parts of this big CAP grant? Sounds like a lot of money, but when you distribute it and you cut the slices very thin after you've got 23, 24 agencies involved in universities. There were permanent research apiaries established to study these things. There were cage and field studies. There were pesticide studies. There were breeding for resistance aspects of the whole project. And the information was then summarized by developing a brand new bee health community of practice on e-extension. E-extension is simply the extension service going into the World Wide Web with research-based information that you can take to the bank. Basically a one-stop shop. So what are the possible causes of CCD? We sat around a table and we banged these out, and then we put teams of people on each individual area to do it. And I think that's the only way to solve some of these really complicated problems. We have existing parasites, mites, and diseases. Is this something we have right now that's causing the problem? Are there new ones coming out? Is it poor nutrition? Nutrition is an underlying factor in many things. We know from our own health. We require a number of different kinds of sources of protein. We have learned that so do bees. Do we have a lack of genetic diversity? Are we having other stressors that are adding to the problem by transportation and migratory bees? Do we have chemical contamination in the wax that the bees are raising young in? Do we have exposure to chemicals for mites that's caused by us? We need to look at these. And obviously, this could be a combination of these factors and others we don't even know about. We know that varroa mite is a serious problem, and I would consider worldwide, most experts would agree that this little parasitic mite that's pinhead size, but looks really big. If you imagine you're the size of a bee, look at that one on the lip of that pupa. Or the size of that mite shown by arrows in the lower right of your picture on a pupa that's been exposed longitudinally in the cell. Mm. It's a serious problem, and such that we really need to do something about managing these, monitoring them, and so we put a lot of effort into that to try to protect our bees from these mites. We know now that additionally that these mites can vector diseases, and including especially viruses. This table, it comes from a Science magazine article from '07, which basically, the purpose of it isn't to just give you a whole bunch of numbers. If you look at the numbers, the disease agents, which are acronyms for Israeli acute paralysis virus, Kashmir bee virus, and the N. apis and N. serrani are meaning nosema, which is a fungal disease. It just pointed out that these are from samples that were supposed to have CCD, and the prevalence of these viruses and nosema were very high. And so that was kind of like saying, we need to study this closer. It's very obvious. If we look at results from the honeybee pest diagnostic lab, and this is, as the information at the bottom, the words say, random. It's from samples sent in to beekeepers from the lab, so it's kind of highlighting those that had problems. But you can see certain things like the yellow line, which is nosema. Its levels are increasing. CCD came about in 05 by the arrow. AFB, which is American foul brood, it's a bacterial disease, is declining. European foul brood is the blue line, it's going up. Tracheomites is going down. And those are general trends that I still think are fairly accurate. So we need to, in other words, we need to worry about some of the diseases in this particular slide. What about the viruses? This was a concerned challenge. And here's a slide showing samples that were taken from the USDA APHIS, National Honeybee Pest and Disease Survey, of virus prevalences. And again, this is black queen cell, BQCB, pretty high, above 80% of those 2013 samples. Also, DWV, deformed wing virus. This is one that you can see in the lower left picture that shows you how the wings of a honeybee are paired, and they normally sit, you know, they're the right size, and they sit right over the back of the bee. But deformed wing causes that wing not to fully develop. So you end up with partial development and deform like the wings of those bees you see in that picture. Some of the larvae may look like the picture in the middle of the lower left valley, but just a sack of virus cells and virus particles. The bees tend to have this greasy, shiny appearance and are kind of hairless on the right. This is particularly probably one from one of the acute bee paralysis syndromes, which is one of these that's been found in samples. We need more study in the viruses. We know that this is a real important area to look at. We don't know, for example, a certain percentage of prevalence is one thing, but what about the virulence level? Some may be more virulent or cause more death than others at a lower level. So we need much more detail on that to get an accurate picture. Well, what are we doing about this thing? And what are some of the effort that's been made so far? Well, the Managed Pollinator CAP, which here's the logo above, is basically was that big CAP grant that got us going. It created an offshoots of a whole lot of different things. It allowed us to form the Bee Health Extension .org website, where now we have upwards of 400 pages of information on that for basically science-based recommendations. We want the beekeepers to use it. I encourage you to use it. It's pretty easy to find site, and please use it. It was made for you. This was largely a result of, I had an IT specialist that was very good with the web. Thank goodness for those people. Michael Wilson, doing a great job. Currently, we have Philip Moore, has taken his place as he's working with the Bee Informed Partnership. Keith Delaplane was very instrumental as the PI of this grant, and Jeff Pettis from the USDA. This is a combined effort from many people to try to get that information up there so that we can all better understand what is going on with the bees. If we look closer at some of these things that are now emerging with the results of the CAP grant and subsequent studies that were stimulated by that one, we know that the existing parasites are still a problem. Varroa is still a problem because it's there. We need to do something about it or it will kill your bees. We do have more tools, and that's encouraging, like resistance and many other things that I don't have time to go into today. We know that viruses are vectored by Varroa, so they move them around. We know that virus can also be transmitted at plants from different species of bees, and that's a concern as well. We know that poor nutrition is emerging as a major problem because if we don't have adequate nutrition and protein, especially for bees, they may be stressed by nutrition. Lack of genetic diversity, we still need more information on that. We know that nosema is present, and some indicates that this isn't a big problem. Others are still concerned, and I'm at a stage where I really don't know because it varies in location. And that's like when you're trying to recommend something to a beekeeper, you can no longer say, well, they all have this. Each colony in your bee yard, if you have 20 bee colonies, or more realistically 10 in a bee yard, all 10 could be different for different reasons. And so now it requires us to know a little bit more. Pesticide exposure is a concern, both due to a very acute toxic exposure, maybe in the field, or nowadays, as we're finding out in the hive, due to chemicals that stay in our wax, unfortunately, that could be sublethal. These effects are very hard to tell, and they're difficult to study. Everybody wants to know the answers immediately and have a quick fix, and there's none available. We just have to keep working on it and keep studying what some of these sublethal effects are, especially when we have these new neonicotinoid insecticides that are systemic, and they're used as seed treatments in many of the row crops. We are studying this right now. That is encouraging. We need to keep doing that. And obviously, to the bottom issue, a combination of all these issues is always possible, and it makes it tougher to describe. Okay, new diseases. And this is something that if you're a beekeeper, you're probably going, yuck, saying out loud. Because you're looking at this and you're going, oh man, does not look normal. This is a relatively, could this be a new disease? It's been around for a long time, but it has been kind of hidden. We've had weather situations and other situations that came that seemed to be very conducive to this particular bacteria, and we had outbreaks of it in the last several years, especially in the South and the Mid-South. One year we lost 25 colonies due to this, when prior to that I hadn't seen it in 20 years. So that was a concern. So these kind of pictures show abnormal conditions that would normally indicate something is definitely wrong. And most beekeepers, when they look at something like this, they know something's wrong and they need to deal with it. We have a publication on our website, and the Bee Health website, that tells all about this. That's where these particular pictures came from. And that's our job. We want people to look at it and understand what's going on. What about some of these pesticide interactions? Everybody's asking about this. This is like, oh, we've got to know right away. Well, we know that we use different chemicals, and the first three that are listed up there are chemicals we put in our beehive. We know that these particular chemicals will be picked up and later will be found in the wax and in the honey if beekeepers have honey on. They know, and they're told explicitly not to treat with these kind of chemicals when they're making honey. That would be a huge mistake. Now, to bring the topic, though, that I want to mention here with the interaction factor is that bees have the ability to detoxify these type pesticides through a cytochrome P450 monooxygenase activity. Or I said that out without stumbling. That's amazing. But the idea is that they have a mechanism to detox this. Okay, but there's only so much limitation that, they can really have. And I'll mention more about that in a sec. Bees, we also know that bees, they're treated with a sublethal dose of coumaphos. We're 14 more times susceptible to apistan. Okay? And so you shouldn't certainly treat with both of those at the same time, or you've got a major problem where pesticides and their effects to a pest are all dose related. And you've got to be very careful with the dose. If you overdose them, you can kill what you're trying to protect. Pre-treatment of bees with fungicides in a study that came out after the CAP grant indicated that this particular fungicide worked in what I call more of a synergistic way, multiplying the effect of crumafloss if the beekeeper were using that, or apistan, tofluvalinate, 72 times and over 1,000 times. So that's not something you want to have in a combination. And we know also that some of these ergosterol biosynthesis inhibiting fungicides, which are big words that say they can get in the way of the detox process, such that now this interferes with the ability of the bees to detoxify these pesticides that they would normally have without having the presence of the fungicide there. Other pesticide-related causes of CCD can be also, and these are the tough ones to get at because they require a lot of study and very careful experiments. Pesticide exposure is one thing, but contamination though in wax, it can be sublethal to the brood. It can also be present in the food at a differing varying amounts because bees store pollen that they collect from flowers from plants, their protein source, in what is called bee bread. They mix it with nectar. It ferments in this kind of like little stack of imagine a little bunch of miniature pancakes all sitting in there that are different kinds of plant pollen. And the fermentation process basically protects it, prevents them from being contaminated and still good, so that the bees go back and forth to their bee bread and get more pollen to basically generate more liquid bee food. Okay? If they go back to get more pollen, and it happens to be contaminated, then they're feeding their larvae with contaminants. These new pesticides are neonicotinoids, and in many ways they're very safe to use for mammals, for us, and for most of our pets. But we're finding out that they're very sensitive and toxic and cause problems to some invertebrates, especially bees, in low doses. So there is a lot of work going on right now in many different venues, both applied and basic, on the effects of these different neonicotinoids, which are systemic. If they are, say you have a plant that has a seed treated, like much of the corn, soybeans, and a lot of the cotton in this country, that material basically goes through the vascular system of the plant, the pipeline, is picked up to where it's in the vascular system, so it protects the plant from injury due to many insects and other things that would cause major problems for that crop. But the concern is that these materials are coming out and coming out and showing up in nectar or pollen that bees would feed on and be subjected to some of these materials. Now, what we don't have is any kind of consensus on what's going on with this. And that's why we are continuing to do studies. We're in the midst of one right now in the Mid-South, looking at row crops and comparing that with other areas that are not around, beehives not around row crops at all. See if we can come up with more about this so we can make be a better informed decision. And we want to be careful what we put on our crops if they have any kind of synergism between what we're using in the beehive for mites. Here's some good news. Well, I guess we needed some after having all this gloom and doom somewhat. And the White House has actually become involved too. This shows you a beekeeper down in the middle where these are the presidential bee colony, okay, as you see to your left. And that the presidential beekeeper, and now they're producing their own White House honey. What I think is great, and I tell beekeepers when they first start out, I don't care if you don't make much honey, you get part of it. And you have a taste of what your bees worked hard and as their keeper, help them to do. But the president's concerned about this issue. We just recently, a number of us went to a summit on bee forage. Prior to that, there was a summit on honeybee health, where experts throughout the country from many, many different agencies and groups put their heads together and try to come up with, and are coming up with things that we need to do to help this whole situation. We need to develop a public education plan. We need to increase integrated vegetation and pest management on federal lands as well. Increase acreage of pollinator habitat under USDA, NRCS, and other programs. Develop pollinator right-of-way guidelines with the Department of Transportation. Create restoration projects through the Department of Defense and the Corps of Engineers. And we need to assess the effects of these pesticides on pollinators. And also make a decision on neonicotinoids, where they be banned, and whatnot. So that's a lot to do. There's a lot of effort that still needs to be done. One thing that I'm pleased to be part of is the Bee Health Community Practice. These are just a few screenshots. If you were in the Washington State County Extension Office and you turned this site up, it would come out Washington State University Extension, because we're partnering in this issue. If you clicked on this particular visual, which live would be simply, would move, then it would go directly to bee viruses and tell you the latest information we have about them. We also talk about our people, who's involved, what they're doing. And like I say, 400 pages of information on bees and beekeeping, and not just honeybees. We have native bee work too. They are very valuable as pollinators as well. The Extension Bee Health website is important too, not only as resources, but also for frequently asked questions. We try to respond to questions we get. We talk about the individuals that are actually doing some of the work. We encourage our colleagues to contribute and continually add more information to this site, and we're doing that. Stay tuned to this site because it's changing every day. We've also had some, it will also summarize, here's another thing that we're doing that I think is valuable. The web is so powerful in that you can get just a nutshell idea of something, or if you want to click on another link, you can go deeper to find the publication that the information came from, or what they're based what they're saying on. And here's an example of this list down below. If you clicked on the Managed Pollinator CAP, the grant we did, we've got numerous articles that were written due to this particular initiative on all these different aspects. We have everything on bee breeding, about varroa mites, about neonic treatments, sunlight, water, and nosema. And you can go and click on these and go right to this publication and get more information on how they did the work. So it shows you the information at several different levels as far as you want to go into it. I think that's very powerful. We've also been trying to respond to the social media, I want to say craze, I guess, for lack of a better set of words, but it is important. People like to learn with things that they can see, and they're visual, and I think it's important. So we've been generating our own videos due to some grant work to the Northeast with different crops, including low bush blueberry and other things, for those of you that are interested in that. You can click on it and learn all about it. You can learn about all kinds of things about bee pests. You can learn all kinds of information. We have over 60 YouTube videos. For those of you that like video learning, we try to make it available for you. And if you've got things you'd like to share, talk to me about it. It has to be research-based, or I won't consider it. If this isn't a political opinion, it's based on science and it's based on research. And that's where we got to keep our focus. I'd like to finish up with a few pretty shots. And no, it's not a honeybee. It's actually a bee on bee balm, which is basically a really good nectar plant. Produces a lot of food for bees. We need to provide plenty of food for bees. You as gardeners, and many of them I'm seeing are gardener related, they're tuning in. The more we can provide food for the bees, the better they're going to do in the future. To help provide these things like cucumbers and squash and other vine crops per se. We're studying also effects of putting these things alternative in really pollinator attractive plants, alternating maybe in rows of a field or along the margins, and using land that would otherwise be wasted as pollinator habitat. And that's part of my own pet peeve and an area which I look at myself. And we certainly have to work together to do this. And I'd like to thank you very much for attending this webinar. And I believe, Sally, you had a comment or two?
Yes, thank you. This was positive in light of a lot of issues. And I do appreciate a couple names I recognized from local NRCS offices. If you would, go ahead and take the poll and ask questions, type them in the chat box. If you, we didn't get to answer them during the webinar. Those of you who have a few more minutes, let me remind you of the additional survey. At this point, I'll open it to any of the other presenters for their comments.
I think we have time for some questions.
Okay.
Hey, John, this is Kathy.
Hi, Kathy.
I see some more questions coming in. But my question is, what can individuals do? What are the best things they can do to help protect honeybee health?
Well, there's probably a whole laundry list, so to speak, but I would say that, you know, depending on what you're doing, if you're a beekeeper, you need the knowledge. Knowledge is very important in what to do about saving your bees. You need to monitor for your pests. You need to know you have varroa mites in your colony, and then you can base your decisions on much more, reliable data. You can't just guess what's in that box. You've got to look inside and see that, and regularly inspect it. You need to make sure you don't treat with any material or chemical when you're making honey. And you need to follow the learning principle, I would say, is join a local bee association, find a local mentor, okay, and go to those meetings and have them teach you. So you have local people there that will answer your questions when that expert's not available. And utilize the web. Utilize bee health. Utilize bee extension, because we have a lot of information to share. If you're a gardener, planting things that bees like. Most flowers are sources of food. Some are not as good as others, but some can be excellent. There's many plants that you can put in your garden that are excellent. A lot of the herbs, salvias for one, sunflowers and many of the family of the Compositae, a lot of the plants in the legumes, which also help your soil. So it's the kind of things that if you put food for bees around your place of residence and your gardens, you'll attract them to your gardens. And not only just honeybees, you will basically attract a lot of native bees. They'll nest nearby, and you'll have sources of pollinators that will help you as well and help your gardens and your crops.
Dr. Skinner, this is Sally Lee. I have a question.
Sure.
In some of the information I've been reading lately, some of the big problems like colony collapse seem to be associated more with commercial beekeepers, those large operations, than the smaller sideliners or hobbyist beekeepers. Has any of your research pointed or focused on those types of arrangements?
Well, in some ways, yes. And I would say, everything has every answer. It's hard to say yes and no to everything. They have a lot of the same challenges. A hobbyist has similar challenges because the bees are bees. However, commercial beekeepers require, since they may have thousands of colonies of bees, they do their beekeeping a bit different. And the hobbyist then may have a little bit of a set of needs that are a little different. However, they all face the same challenges, and they all need to look at their bees and make sure they know what they have in those hives. The more experience they have, whether they're in, most of the commercial people do, or they wouldn't be in business. So some of them are generally more experienced than you might say in a hobbyist. But I certainly wouldn't want to slight a hobbyist at all, because more beekeepers in the United States are hobbyists than there are commercial beekeepers. The commercial beekeepers run many colonies of bees. That's true. So they have certain needs and requirements that hobbyist that only has 5 to 10 colonies of bees in their backyard, or 1 or 2, they don't have the same needs. But both need to go out in their beehives. Both need to understand the biology of the bee. They need to understand what symptoms from some of these different diseases and pests are, and what to do about them. And that's why we try to provide that kind of research-based information on strategies for those beekeepers to keep their bees alive. We also need to realize, too, that in different regions, we may have a little bit different challenges due to forage, maybe due to local weather constraints. Some areas of the country may have a more challenge with drought than others. So therefore, their foraging resources are maybe limited compared to others that have plenty of rain, or maybe too much rain. So it's a case of where nowadays even the hobbyist beekeeper is still a challenge, because their bees may be bee colonies in one apiary may be very different from one another. And trying to keep ahead of all the challenges. Not to say that you as a beekeeper are going to find all those things in your apiary. Probably doubtful. But still, you need to have the knowledge to be able to figure out, well, number one, something's wrong. And then, what do I do about it?
Okay, thanks.
Sure.
Okay, John, we've got a number of questions in the chat box. So, Sally, perhaps you and I can sort of alternate here and get these covered as well. I'm going to start with a first question was, are your studies focusing on commercial bee operations only?
No. That's one of the few times I can say no. I mean, we in Tennessee, for example, we are basically a hobbyist state. We have a number of sideliners, which I define as having more colonies than a hobbyist. But I'd say there are some other people are concentrating more on commercial beekeepers, and that is important. There are others of us that deal with our own states, our hobbyists, our local associations, and we try to educate by doing that way, by state associations and local associations, which are very important. And I'm not trying to underestimate the importance of that commercial beekeeper either.
Someone else asked, where did the grants come from? I think you covered that partially. You mentioned USDA.
Right. There are a number of agencies that have grants that researchers, in combination of teams mostly, because we're finding that these are complex issues, and it's not just one person going to be able to solve the problem. So often we put teams of people together to look for grant monies. These can be from the USDA, from the National Institute of Food and Ag, NIFA. They can be from other scientific organizations like NSF. Under NIFA, there are a number of different groups, especially Crops Research Initiative, and other ones. The EPA has some funding available to study aspects of these things. NRCS, under basically USDA, that they are very involved in this thing. And there are other foundations out there. And basically, we have an increased amount of funding that has become available, and we're trying to actually do the best we can with those funds, make sure they go as far as possible.
Okay. Another question relates to plants to use as pollinator food sources, and I think someone mentioned there are regional lists. There's one here for the southeast, and there's probably several more for northeast, northwest, southwest. Xerces has a good list for native pollinators by region, and that's just the beginning.
That's right. And a lot of these groups were available. I was just at a meeting after the task force met. I went to the North American Pollinator Protection Campaign, where a lot of these different agencies and entities were there. NRCS has all kinds of information. If you go also to pollinator protection and you look at that website, you can find links to these different list guides that were made in cooperation with NRCS, Xerces, and other groups to come up a list of forage plants that are regional guides. And they're by different biome as well. So not just necessarily a specific region, but might be relative to soil type or other soil designation or other things that we know are important. And those can be taken advantage of. Xerces has several publications out that are very valuable and should be looked at. And if you were to Google resources and forage resources for bees, you'd come up with a lot of information. I would suggest you look especially hard at the ones that are research-based information, because not all lists they're easy to make a list, but I'd rather make sure that if I planted that list, that it would actually come up and they would be compatible with one another. But it does give you an idea of what you could pick out of that list that you would like to put in your backyard or next to your garden. So that's important.
There's another question that's of interest. Any chemical we can use to prevent the loss of bee colonies?
Well, that's an interesting one. I just don't have enough information about it to know exactly what their thoughts were on that. I mean, we can use particular kind of chemicals to basically combat mites or diseases if you're a beekeeper. And these don't have to be all, they can be organic, they can be inorganic, they should always be used according to a label precaution, not just something that is cooked up in somebody's bathtub. I mean, we really have to be responsible that we don't abuse chemistry. Consider the dose response always. Something in a very low dose might be a medicine, but in a higher dose it can be a toxin. And so it's hard to say where that, exactly where they're going with that question. I guess I'd need to have something a little bit more specific.
John, we have a question about, are there geographic areas of the United States where CCD is more critical in terms of economic impact than others? Or I guess I should say honeybee decline is more critical. Are there some areas harder hit than others?
Well, historically, if we look at that situation, when we do have probably the data, I would like to look at something more current that comes out of the Bee Informed Partnership, because they're generating information from beekeepers all the way across the country to just look at that issue. I don't have that right in front of me right now, but I will say historically that, for example, in Tennessee, when in the early right after CCD was basically discovered, we didn't see those kind of symptoms in our beehives. Currently, I get a few people every year that think maybe they've got this, and maybe they do. We have to look at each one of those situations individually. I would say the Northeast seemed to be hit very hard because that's where the initial person came from, Pennsylvania. But they were also a commercial beekeeper that moved their bees up and down the East Coast for pollination. And we know that, there were colonies in the Pacific Northwest and in some of these areas that will also seem to be affected. Probably the middle parts of the country, to me, it didn't seem as much, but I don't want to speak for everybody else's place by entire region. Good question, though.
We had another question or a comment that someone would like to see some more information about the neonicotinoids impact on honeybees. Is that article, a Xerxes Society article, a good place to start? If so, I can paste that link into the chat box. Or do you have another suggestion where someone could start reading to get some science-based information?
Well, there's a lot of information out there, and you do have to be careful about that. I guess that's the kind of thing we need to do, and so I'm going to get on it. All I can say is we are currently, with our bee health site, what we have done is we summarize the information we know currently about varroa and viruses, and a lot of that's as current as we have. But new data is coming out every day. New studies. It's a very hot area, and I'm glad because we need all the information we can get. So we have plans to do a summary paper basically on and like a review of these neonics as this information comes in. So I can't say I've got it right now. I've got stacks of things on neonics, but any one of them I don't know that I could pop out and say use this one as your key. That would take a little bit more conversation. Maybe that person should contact me directly.
Okay. Georgianne asked, I participated in a webinar a few months ago, and the point of view was presented in the webinar that GMO plants were the primary culprit. Is that opinion being discounted in view of this variety of problems?
Okay. I think I got that question, but my comment on that is that, and this is based on sitting down with a whole group of people, scientists, trying to come up with what do we think are the causes for some of these problems. And we listed them and then we prioritized them. And the GMO issue was put down as the dead bottom line for everybody in the room, without any doubt. And yet as a scientist, I'm still a bit of a skeptic, so I've got to look at it and consider that there still needs to be looked at. But most people would discount the role of GMOs in this particular situation in colony decline. I know that brings up, people that have strong feelings. What I try to do is keep my emotions out of it, base it on science, and I don't see the science to back up that it's a problem.
You don't see the science to back up that there is a problem with GMOs, just to
That's
clarify.
Correct.
Okay.
That's right.
Here's an easy one, John. How many bees are there in a honeybee colony?
Well, gee, it depends on when you're talking, what time of year, but you start with a package of bees, and if you're going to put a package of bees brand new into a colony where they are 3 pounds of bees, and so you've got about, so you're starting with probably, you do it by weight of the pound of bees, actually, believe it or not. And so you're probably starting with maybe 10 to 15,000 bees, and then they can build up and during a season with productive food, and especially as they get bigger and bigger in the second and third year that they're in existence, to where you've got colonies from 60,000 to 70 to 80,000 and above, and that's a lot of bees in the boxes. Are there other questions?
And is anyone who has a few minutes not responded to the survey on bit.ly? If you would, please, that would be great.
All right. And we have some more questions here. Let's see. Lots of questions. It's hard to make sure we're asking them all. Effects of common pesticides available to home gardeners on bees. I think if you go to that Xerxes Society link that I pasted in, it will talk about some of the neonicotinoids that you may not realize you're buying as a homeowner as well.
I think that would be a good source to go to. I agree with that.
All right, let's see. There is a recent paper that came out, and there was Kevin Wanton would like to see a study of neonicotinoid landscape insecticides comparing dosage with ag-related insecticides. And again, that Xerxes Society article talks about those differences in doses from what is in the landscape versus what's in the agricultural field.
Yeah, that's a good question.
Yeah.
And I do think we need to take that into consideration when we're considering, as gardeners, we're considering about treating things. And there are availability of neonicotinoids and other insecticides that aren't under regulation that that could be a concern. Mm-hmm. I don't know that we have a good handle on how much of those materials are being applied by homeowners and gardeners. And maybe we should develop cautions and best management practices on that in the future.
All right. Jeannie, do we have a few more minutes because we have a few more questions, or do we need to sign off?
You can have a few more minutes.
Okay. John, if you're up for it, here's another one. Is there associated or adjunct study regarding changes to food grade honey when stages of colony collapse disorder are identified?
Well, if I'm hearing the question right, this is what they're looking for, is like, does the fact that you have CCD, is that affecting the quality of honey? And I would say not necessarily at all, because if the beekeeper is reading the label properly, if this is concerning chemicals, then they shouldn't be treating when the bees are making honey. And so if it's, that's one time period where beekeepers know, whether they're commercial or hobbyists, that if they treat with certain chemicals, that chemical can go in the honey, and you would not want that to contaminate your honey. So that's a big concern that we really make sure that beekeepers understand that. All the classes that I teach and anybody else that I know, we really make sure they understand, do not treat while you're making honey. So that's kind of like a major rule. And I don't, and again, I'm not aware that would be a problem.
What about mosquito control pesticides? This comment is there are commercial providers spraying a lot in this particular city.
I see. Well, that can be a concern depending on what they're spraying. And they should, technically, and it would depend on your locality. There are some local entities would basically have provisions to inform all the beekeepers when they're going to spray, where the location is, so that they can basically either confine their bees or move them to avoid having any problem with any insecticide they're using for mosquitoes. And I don't really think that should be done. And it's probably too bad it may not be done everywhere. But I know in some locations it is, and that they've been successful to reduce problems with mosquito spraying and any kind of toxic problems caused by that. It also behooves your beekeepers, if you have a state, regulatory inspection service, and it requires you to register your bees, that you actually register them. They have a list then of locations, and they know where you are, so that maybe they can actually send something out and warn you that we're having a spray in this location. You need to take action to avoid having a problem.
Any more questions? This has been great. I'm going to go out and check our hives here at the Botanical Gardens right now as soon as this is over.
Well, if it's above 55 degrees and it's decent weather, then you can go for it. But if it's not, be careful because you don't want to chill that brood.
Oh, no. I will not open the hives. I'll just see if they're out and about.
Okay. Well, that's if you got decent weather, and they should be flying in and out regularly, and hopefully maybe in your neck of the woods they might be bringing back pollen. That's a good thing.
Yeah. Thanks, everyone, for attending. If there are additional questions, you've got Dr. Skinner's contact information. And from the pictures on the internet of him in his bee suit, it looks like he could answer most anything.
I can try, or I can give it to somebody else to answer.